In this video, we'll be looking at how to do a mono and dihybrid crosses. Now, obviously, these are the simplest type of crosses you can do when you're reading genetic crossing problems. But we need to, before we do that, as you can see, we need to define two terms that are important to know when you're crossing these kind of genetic makeups of organisms. And that is heterozygous and homozygous. By the way, I won't be focusing on the biology of genetic crossing, like Mendel's 3 laws. We'll do that in like other videos. But right now, I want to show you the arithmetic of actually crossing these genotypes, right? So heterozygous, right? A genotype that carries different alleles, which means it has one dominant and one recessive. So this is also called a heterozygous, is also called a hybrid, in case you didn't know. So for example, big T, which represents dominant, and small t, which represents recessive. So that is a heterozygote because it carries two different alleles, and alleles are just traits that are traits with variations. That's what an allele is, right? Traits with variations. Now, homozygous, on the other hand, as you could probably imagine, is different from that. It's when an organism has the same allele. So it could be TT or small tt, big TT, which these represents a recessive genotype and a dominant genotype. So they're just organisms. They're just genotypes that carry the same alleles. So if it has one recessive allele, then it will have another recessive allele. They carry the same type of alleles. And now that you know that, we can further get into the main point of the video. So those themes are very important when you're reading problems. And yeah, how to do a monohybrid cross. Now, let's say I have big A, small a times big A, small a. This is dominant, recessive, and I wanted to cross this. Well, what I would do in this case is I would make a 2x2 box, and I would organize it like that. So I have it organized like that, and obviously make sure the dominant trait always goes first. Now, how you do this is, as you can imagine, you just put the terms together, so the letters together. So this A goes with this A. So this is AA. This A goes with this A. This is A. This is then this A goes with this A. This A goes with that A. So you literally just match them. This is just pattern matching from that point. So obviously, this is a very simple and very basic cross. And so we'll talk about genotype frequencies, allele frequencies, genotype ratios, phenotype ratios in later videos, but I want to just cover the aspect and a very, very simple concept in genetics before we actually start getting into more advanced videos. So you know how to do a monohybrid cross. Now let's look at dihybrid cross. There are two ways to do a dihybrid cross. One, you make two monohybrid crosses, or another way, you just do the actual way. So the first way I said, maybe you make two monohybrid crosses, is kind of a cheat code. But here's the thing: when you make two monohybrid crosses, you can only look at the genotypes of those organisms. Rather, if you do the more orthodox way, which is essentially making this 4x4 box with 16 squares inside of them and crossing them, you can look at the genotype and the phenotype. Right? So like I said, when you only do two monohybrid crosses, you will only be able to look at the phenotype. So yeah, you'll only be able to look at the phenotype, so you can't look, if the question asks, oh, give me the exact genotype, you won't be able to do that. So let's actually get started. So I'm just going to be doing two hybrids. So let's say we have T times, two hybrids. TT times AA times TT. we will be doing two of the methods that I just explained. We'll be doing, first of all, we'll be doing the more orthodox method, which is essentially what you do is, it's also important to understand the law of independent assortment and the law of segregation and the law of dominance, because when you understand those, this will be very simple, but I want to get into that later in the channel. So what you need to do is you need to cross this A with that big T. So this becomes AT. This can then cross, and then once you do that, you need to cross this A again with the small t, and you write it down. And then you do it with the same letter, A, T, same letter, A, T. And you do, and I made a very easy one because, oh, these are very uneven squares. Just ignore it. That's so uneven. Like, what is this? But yeah, that's actually extremely uneven. Yeah, let's just make. So I actually made this question very easy because, as you can see, they have the same genotype. So I can just rewrite this again. You know, that was kind of cheap because I'm too lazy to actually like use my brain. So now let's just write AT, 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 AT. Now that you know that this goes with this, and then this goes with that goes with that, and that goes with that. So you're just taking this letter and distributing it with this one, and then taking this letter, distributing it with this one and this one. So now it's the same concept as a monohybrid cross. You just put the letters together, and remember the dominant letters always go first, right? Capital dominant letters. So this would be A T T A T T. I'm gonna write just all of these down. Make sure the same. Make sure you don't write it like A T A T. You don't want any of this. You want to keep the letters organized by, you know, the same letters. So A T T A T T, and etc. I'm not going to finish it off because it's not like we're looking at any genotype or phenotype ratios, but you understand the concept or the methodology to doing this, right? So now you understand it. Let's look at method 2. You can finish this off if you want, but essentially you understand you're just putting it together and you just distributed the traits. Now let's look at a little less orthodox approach, a faster method. But like I said, way two is only important. You can only use way 2 if the question asks for the phenotype. If it asks for the genotype, then you can't do way 2. So like I said, I think I said way 2 or way 1, but whatever. But just the less orthodox approach is making two monohybrid crosses. And what you do is you literally just take this genotype right here and you're like, yo, I'm gonna just cross this right now. And then you just do that. So did you see? Can you analyze how you just took this part and crossed it with this? And you literally just crossed it with those two. And then you're gonna, as you can imagine, you just do the same thing here. Take that part and cross it with that part. So just take both genotypes and cross them. And then with this, you can it's obviously faster because doing monohybrid crosses are faster and you're less prone to make a mistake. So you just quickly do that, 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 quickly do that. So now that you know how to do both methods, I think you'll be successful in solving genetic crosses, but with obviously constraining to the fact that we only learned mono and dihybrid crosses. We'll learn more advanced crosses, but essentially they have the same concept as these crosses. So these are really the fundamental of genetic crossings. Now, like I said, the more orthodox approach, you will be able to analyze the genotype and the phenotype. In that less orthodox approach, meaning the less traditional approach, this approach, you can only look at the phenotype here, right? You can't look at the full genotype. You can only look at the phenotype here, which is since you can only look at the phenotype here, if the question asks, like I said earlier in the video, if it asks for the genotype, then this wouldn't be a great method to use, and you would have to use the first method, the more orthodox method. But if they just ask, oh, find the phenotype, what is the chance this will have this phenotype, then you can use the less orthodox method. I know that sounds a little confusing. I will be putting a description in the video so you can read that kind of the kind of phrase I just said a little bit more clearer. But yeah, that's going to do it for today's video. Stay tuned for more videos. We will be posting every week, and this is just the beginning and basics of heredity, and you have a lot more to learn. And I'm no pro at this, but I'm just I'm not even that good. I'm just literally doing these videos, and stay tuned for the next video.